All right, ready for deep dive number 8, BiochemSerye Learning on the Go. Today we're tackling peptide bonds.
Oh yeah, peptide bonds. The glue that holds proteins together.
But we're also going to figure out how they can be broken down too.
It's all in here, from textbook chapters to video lectures.
Your express lane to understanding these bonds. You ready?
Let's jump in.
Before we go too far though, let's get on the same page. What exactly is a peptide bond?
It's the connection you make when you're linking amino acids, like snapping together Lego blocks. It's a special type of bond.
And a bond, right?
Exactly.
Okay.
Formed between the carboxyl group of one amino acid and the amino group of another.
So you can just keep snapping on more amino acids onto either end of the chain, right? Like Legos.
Absolutely. But, and this is important, peptide chains have direction.
Like a beginning and an end.
Yeah, just like reading a sentence. You start at the amino terminus, the N-terminus, that's the end, with a free amino group.
And then go to the carboxy terminus.
Right, or C-terminus, that's got the free carboxyl group.
So if we're talking about angiotensin II, you know that hormone that regulates blood pressure?
Yeah.
We always write that sequence starting with the N-terminus.
Precisely. It would be asp arg valtir il his prophy, from N-terminus to the C-terminus.
Like a biochemical sentence.
Exactly. It's got a specific order. It tells us how they're all linked together.
This is where I tell my students, think of it like a string of pearls. Each pearl is an amino acid and the string that's connecting them.
That's the peptide bond.
Right.
I like that visual. Simple, but helps you understand.
Okay, so speaking of connections, let's talk about why they're so strong and stable.
That's where resonance comes in.
Oh, yeah. Resonance.
I have to admit, resonance really made me think when I was learning it.
It's one of those aha moments in biochemistry for sure.
Yes.
Okay, so when we draw a peptide bond, you see a double bond between a carbon and oxygen and a single bond between a carbon and a nitrogen.
Like the textbook picture.
Right, but it's way more dynamic than that.
Huh.
Those electrons in the bond aren't just sitting there, they're moving around, creating a cloud almost.
Yes, an electron cloud that's shared between the carbon, oxygen, and nitrogen.
So instead of a rigid double and a rigid single bond, it's more like they're sharing the double bond character, which makes the structure more stable.
Precisely. And this stability, that's huge for protein structure. The resonance forces it to be planar.
Meaning all the atoms are lying in the same plane.
Exactly, flat plane.
So instead of a flexible string, it's more like a rigid plank connecting those Lego pieces.
Yeah, and that rigidity changes how the whole protein chain folds and twists, like a choreographer, setting specific limits on a dancer's moves.
So we've got our strong, stable peptide bond holding those amino acids together. But how do they actually form? It's like a tiny construction project.
It is, and every good project needs a process. And this one's called dehydration synthesis. Picture two amino acids, they're coming together to bond. One has a carboxyl group and the other has that amino group.
Right, the ones involved in making the peptide bond.
Exactly. So during dehydration synthesis, a hydroxyl group, OH, is removed from the carboxyl group.
And at the same time, a hydrogen is removed from the amino group?
You got it. So we're pulling out a water molecule from those two amino acids.
Hence the dehydration part.
You got it. And then as that water is formed and released, the carbon of the carboxyl group and the nitrogen of the amino group form a bond. Bam, peptide bond.
It's like those little construction workers are using the water as the mortar to connect the bricks.
Huh, I like that.
But wait, shouldn't we talk about the charge of the amino acid? Something about a zwitterion?
Oh, you're right. We can't forget those zwitterions.
Okay, remind me again, what's a zwitterion?
So at physiological pH, the pH of our cells and our body, amino acids exist in this special form. The zwitterion, it's like they have a split personality.
Positive and negative charge.
At the same time, the amino group, it picks up an extra hydrogen ion, becomes positively charged, NH3 plus T.
And then the carboxyl group loses one.
You got it. Becomes negatively charged, COO.
Like a magnet with a plus and minus pole.
Perfect analogy. And this form, this zwitterion, is super important for peptide bond formation.
How so?
Well, the negatively charged carboxyl is attracted to the positively charged amino group of another amino acid, brings them close enough for that dehydration reaction to happen.
So those charges are helping the workers line up the amino acid bricks just right before they cement them together with a water molecule.
Exactly. It's a great example of how the chemical properties of these amino acids affect how they interact.
And ultimately lead to those essential peptide bonds.
The foundation of all proteins.
This is all so fascinating. But can those bonds rotate freely? Can they just twist and turn?
That's a great question, and it takes us to another important concept. Restricted rotation around peptide bonds.
Right.
Remember that resonance stabilization?
The planar structure.
Yep, the delocalized electrons. That puts a limit on how much that bond can actually rotate.
So they can't just spin freely.
Not really. Think back to those Lego blocks. The plank connecting them has to stay flat. The atoms in the peptide bond are fixed in that plane. They can rotate around the bonds connecting them to the rest of the amino acid, but the bond itself is rigid.
Got it. So they've got some wiggle room, but there are limits.
Exactly. And those limits, they have a huge impact on the final shape of the protein. Like, imagine building something with Legos if you couldn't rotate any of the connecting pieces.
You'd be pretty limited.
Yeah, just straight lines. So to get a little more scientific, we describe these limitations using what we call dihedral angles.
Dihedral angles?
Yeah, specifically phi and psi and as. These angles describe the rotation around the NC and C bonds.
Okay, hold on. NC and C bonds. Can you break that down a little? Some of our listeners might not be familiar with all the technical terms.
Sure. Think of the amino acid like a little branching structure.
Mm-hmm.
The N-C bond is the connection between the nitrogen atom of the amino group and the alpha carbon. We call that C. And the COC-EC bond is the connection between that alpha carbon and the carbon of the carboxyl group. So these dihedral angles are describing how much those branches can rotate around those specific bonds.
So phi and psi.
Mm.
Are there certain values that are allowed? Are there some that are forbidden?
Exactly. And that's where the Ramachandran plot comes in.
The Ramachandran plot.
Yeah, it's a visual representation of all the possible combinations of those phi and psi angles. Think of it like a map. It shows the allowable pathways for that peptide chain to fold.
So it's a guide for how they can twist and turn while still staying within the limits of those rigid peptide bonds.
Exactly. The green areas on the plot, those are the combinations that are sterically allowed.
Meaning the atoms won't bump into each other.
Exactly. And then there's red areas. Those are the no-go zones. That's where the atoms would clash.
So it's like a choreographer marking out the stage for the dancer, showing them where they can move and where they need to avoid collisions.
I like that. It's amazing how these limitations, they actually lead to elegant and complex protein structures.
They do. So we've talked about how peptide bonds are formed, but what about breaking them down? Our bodies are constantly breaking down and rebuilding proteins, right?
All the time. Like a molecular recycling center.
So how do we break those tough bonds?
It's called hydrolysis. Remember how we formed them? We removed a water molecule during dehydration synthesis.
Right.
Well, hydrolysis is the reverse. Add a water molecule back in, splits the bond, and we get those individual amino acids again.
Like adding water back to the mortar to break the bricks apart.
Exactly. Controlled demolition at the molecular level.
So simple, but so important. Dehydration to build, hydrolysis to break.
Fundamental to how our bodies deal with proteins.
It's a beautiful molecular dance. Peptide bonds are definitely the stars of the show.
Absolutely. And that dance is happening in every cell, all the time. Essential for everything. Growth, repair, keeps our system in balance.
So we've covered a lot, but I think we've only just begun to scratch the surface. Before we dive deeper, let's do a quick check. How much have you absorbed? Ready for some rapid-fire questions?
Let's do it.
All right, question 1. What kind of bond links those amino acids together? Think back to the Legos.
A peptide bond, a special kind of amide bond. It's the link between the alpha carboxyl of one amino acid and the alpha amino of another.
Perfect. Question 2. What's the big deal about resonance in a peptide bond? Remember those zipping electrons?
Resonance means the electrons are delocalized, gives it that partial double bond character, makes it stable and planar. Both super important for how proteins work.
Right. Question 3. How is a peptide bond formed? Remember our little construction workers?
Dehydration synthesis. Remove a water molecule, take the OH from the carboxyl group and H from the amino group. Water's released and boom, peptide bond formed.
All right, question 4. How do we describe the direction of a peptide chain? Think biochemical sentences.
We always start at the N-terminus, that free amino group, and read through to the C-terminus, the free carboxyl group.
Great. Now for question 5. Why is there restricted rotation around that CN bond in a peptide bond? Remember that rigid plank?
Because of resonance. That CN bond has partial double bond character, and that limits rotation. Super important for maintaining the shape of proteins.
Exactly. Question 6. What are dehedral angles, those valesive, and why are they important? Think about those restricted dance moves and those bonds within the amino acid.
Those angles describe the rotation around the NC and C cobonds, and they ultimately determine the different shapes the peptide chain can take, which contributes to the overall 3D structure of the protein.
Awesome. Question 7. What about the Ramachandran plot? What does that show us?
It's a visual representation of all the allowed combinations of those half-in angles. Basically, it shows us which combinations work without atoms bumping into each other.
Like a map of allowed dance moves. Okay, question 8. How do you break a peptide bond? Think demolition.
Hydrolysis. Add that water molecule back in, break the bond, release the amino acids, the opposite of dehydration synthesis.
Got it. Question 9. What's a zwitterion and how does it relate to peptide bond formation? Think about those magnets.
A zwitterion is a molecule with both positive and negative charges at the same time, but overall neutral. Amino acids at physiological pH exist as zwitterions, and this form is important for peptide bond formation because those opposite charges attract each other, bringing the amino acids close enough to react.
Awesome. And finally, question 10. Why are peptide bonds important in biology? Think big picture.
Peptide bonds are the building blocks of proteins, and proteins are involved in pretty much every process in our bodies. Without peptide bonds, we wouldn't have the amazing diversity and function of proteins that make life possible.
That's a fantastic summary. I hope you all enjoyed that rapid-fire Q&A, but we're not done yet. In the next part, we're going to dive even deeper into how those unique properties of peptide bonds affect protein structure and function.
Whew. That was a good warm-up. You know, it never ceases to amaze me how much information we can pack into a quick Q&A. But there's still so much more to explore with peptide bonds.
We've laid the groundwork, but now I want to build on it. So we've talked about these restrictions on rotation. But how does that actually play out in the real world of proteins?
Imagine if those peptide bonds could just rotate freely. Proteins would be these floppy, random chains. No way they'd be able to fold into those precise 3D shapes. And those shapes, crucial for their function.
Right, like those limits on a dance floor. That's what allows the dancer to create those beautiful, specific moves. So how do those restrictions actually guide protein folding?
It comes down to those phi and psi angles. Remember, they describe the rotation around those specific bonds in the amino acid structure. Because of the limitations of that planar peptide bond, only certain combinations work.
And that's where the Ramachandran plot helps us.
Yeah.
It shows us those allowed combinations, like a map.
Exactly. And as that chain starts to fold, those allowed angles, they kind of dictate how the amino acids can interact. Some combinations will lead to alpha helices, those spirals.
Right.
And others will lead to beta sheets, those flat structures.
So it's like those restrictions are actually driving the formation of those classic secondary structures we see. It's not limiting, it's creating possibilities.
Exactly. Constraints can actually lead to more creativity.
I like that.
So let's zoom out for a second. Think about how those secondary structures, the helices and sheets, come together.
To form the 3D shape of the protein, the tertiary structure.
Right. And that's where things get really interesting. All sorts of interactions are happening between different parts of the chain. And those interactions, that's what stabilizes that final 3D shape.
So it's not JC about the peptide bonds anymore. There are other forces at play.
There are. You've got hydrogen bonds forming between different amino acids, ionic interactions between charged groups, hydrophobic interactions where the nonpolar ones clump together to avoid water,
Right.
and even the sulfide bonds, those are strong covalent bonds, between sulfur atoms and cysteine.
Wow, so many different forces. It's like a symphony, isn't it, to create that masterpiece?
It is. And all of them are influenced by the sequence of those amino acids. That sequence dictates how the peptide bonds are arranged and how the protein folds.
Which brings us back to that primary structure.
Yes, that linear sequence, like the blueprint for the protein.
Okay, time for some real-world examples. Think about collagen. Tough, fibrous protein. Skin, tendons, bones.
Oh yeah, good one.
Made up of those long triple helix structures, like 3 ropes intertwined.
And those helices, stabilized by hydrogen bonds between specific amino acids. Without those, collagen wouldn't have the strength and flexibility it needs.
Right. And then there's hemoglobin, the one in our red blood cells. Carries oxygen.
But hemoglobin, complex quaternary structure, right?
Yes. 4 subunits, each with a heme group. Binds oxygen.
And the interactions between those subunits, they're crucial for hemoglobin to bind and release oxygen just right.
Structure and function, always linked. So we've gone from these tiny peptide bonds all the way to complex protein structures essential for life.
It really is amazing. Those limitations on bond rotation dictated by resonance, they actually set the stage for this incredible diversity, all these different forms and functions.
It really shows the elegance of nature's design. So what about breaking those bonds? We talked about hydrolysis,
Mhm.
but where does that actually happen? It's not just random, right?
You're getting at another level of control. Hydrolysis of those peptide bonds is done by very specific enzymes,
Enzyme.
proteases. They're like the demolition crews of our molecular cities, breaking down those proteins that are damaged or not needed, or even from the food we eat.
Makes sense. It's like an organized recycling program.
Exactly. And the specificity of these proteases is amazing. They recognize certain amino acid sequences and they cut the peptide bond in very precise locations.
Like a molecular blueprint. They know exactly where to make the cuts.
Yes. And this is super important for regulating tons of cellular processes, protein turnover, signaling, immune responses, even programmed cell death.
Wow. So dysfunction in that protease activity could be really bad news.
Oh yeah, all kinds of diseases, cancer, Alzheimer's, infections.
It's amazing how these tiny peptide bonds and those enzymes that break them are involved in so much. Our health, our well-being. We've talked about how they're formed, how their rotation affects structure, how they're broken down. But can we manipulate those processes? I mean, can we use this knowledge to design new drugs, even new materials?
Now you're thinking like a scientist. That's where we get into biotechnology and bioengineering. Remember those proteases? Researchers are working on something called protease inhibitors, molecules that block their activity. They could be drugs for all sorts of diseases.
So it's like a molecular wrench that can jam the demolition crew.
Exactly. And in material science, they're looking at the strength and flexibility of proteins, especially ones with repeating amino acid sequences, trying to design new biomaterials.
Like spider silk.
Yeah.
Strong, lightweight.
Perfect example. It all comes down to those peptide bonds and how the amino acid chains interact. And researchers want to mimic that to create new materials for all kinds of things. Wound healing, tissue engineering, even bulletproof vests.
Wow, who knew? From the basic building blocks of life to cutting-edge technology, all thanks to these peptide bonds.
It's incredible, and it shows the power of scientific research. Understanding these basic principles can open up so many possibilities.
It's a journey, that's for sure.
It really makes you think. Those tiny little peptide bonds, just a simple dehydration reaction, but they're the basis for all these different proteins, all the functions in our bodies.
And it's not just that they're there, it's how they behave. The restricted rotation, that planar structure, the specific angles, it's all crucial for how proteins fold and how they work.
You know, sometimes I feel like I'm explaining this to my students for the 100th time, but then I remember when I first really got it. Peptide bonds, I mean.
When was that?
It was during my postdoc. I was working on this enzyme. It was giving me a real headache.
What was the problem?
I was trying to figure out why this tiny change, just one amino acid, totally changed what the enzyme could do.
Interesting.
Yeah.
What was going on?
Well, it turns out the mutation messed up a hydrogen bond. It was supposed to be stabilizing a loop near the active site. That loop was important for the enzyme to, grab onto its substrate. And without that hydrogen bond, the loop went all floppy. The enzyme just couldn't work right.
So the peptide bonds themselves weren't changed, but messing with the amino acid sequence messed with the interactions between them.
Exactly. That's the key. It changed the whole structure, the whole function. That's when I really saw how important those basic principles are. Peptide bond geometry, the forces that hold those proteins together, how specific the interactions are between those amino acids.
It really shows that proteins aren't just static, they're dynamic. They're always responding to their environment, changing their shape to do their job.
Absolutely. And that dynamic nature is essential for life. Think about how enzymes work, how antibodies grab onto things, how muscles move. It all depends on proteins being able to change their shape.
It's a very specific way, too.
Exactly. It's like a dance. All those proteins twisting and turning, connecting and disconnecting, all in response to what's going on around
A
them.
beautifully choreographed dance.
That's a great way to put it. And it's happening in every cell all the time, a symphony of movements. And it's all thanks to those peptide bonds and the forces that control them.
So we've covered a lot of ground today, from the basic structure of peptide bonds all the way to their role in protein folding and function. And we even touched on how we can use this knowledge in medicine and engineering.
Hopefully our listeners have a new appreciation for these molecules. They're complex, but they're also elegant, and they're vital for life.
It's amazing how something as simple as a bond between two amino acids can be so important. It shows that even the smallest things can have a huge impact.
It all comes back to scientific inquiry. By understanding those basic principles, we can do so much. Treat diseases, design new materials, even engineer new life forms.
Wow. So that brings us to the end of our deep dive into peptide bonds. But don't stop here. There's always more to learn about these fascinating molecules. Keep asking questions, keep exploring, and never stop learning.
Absolutely. And if you ever find yourself wondering about proteins, remember those tiny peptide bonds. They're holding it all together in that beautiful, powerful dance of life.
Thanks for joining us on this deep dive. Until next time, happy learning.